So the next topic that we have now is of fertilization. Till now we had seen that when we see the reproduction and development in angiosperms, we broadly classify the entire event into 3 stages, and one of the stages was that of fertilization. This is the important stage where male gamete, which is formed as a result of formation of male gametophyte and female gamete, they are going to fuse together to form the zygote. Alright. Now, this male gamete lies within the pollen grain, and before this lesson we had talked in great details about the process of pollination, where the male gamete, which is within the pollen grain, comes and reaches the stigma and has to reach the ovules inside the ovary, wherein lies the embryo sac that houses the female gamete. This is just a general representation. This is the female gynoecium, as you can see within the flower. The pollen grain in the 2-cell stage or the 3-cell stage, depending upon the type of family to which the flower belongs, it would come and fall upon the stigma. Now this stigma has chemoreceptors in it. What happens is, what enables the pollen grain's gamete that lies inside it to reach to the ovule, which we have studied the structure of an anatropous ovule, what it looks like. It has a micropyle. The gamete has to enter from this region to the micropylar region or chalaza, wherever the entry is enabled. That region is to be reached, and that is done by formation of a pollen tube. Now, whenever this happens, that the gamete is being carried by the formation of pollen tube through the style after reaching the stigma, being receptive to the given pollen in order to reach to the ovule, the pollen tube is being formed as we have talked. So this formation is known as siphonogamy. Now we are nowhere talking about the fertilization that would give rise to the fusion of male and female gametes. Before reaching that, we have to understand this term. This is important, that what is siphonogamy? Siphonogamy is the event in which the pollen grain, on reaching the stigma, which is receptive to it, forms the pollen tube. Now this pollen itself does not move through the pollen tube. It is the pollen tube that is being formed by eating up the tissue of this particular style. So there would be formation of a tunnel-like structure within the style, because the pollen grain would lead to the formation of pollen tube by releasing pectinases and hydrolases, which are, of course, the name is suggesting that they are enzymes. Both these enzymes, they eat up the tissue that is present inside the style. As soon as they eat up, there is a passage that is being formed and the pollen falls down. Okay, this particular event is chemically guided by calcium boron inositol complex. So you remember when we talked about the importance of boron as a micronutrient in mineral nutrition? It was an important compound. It was an important element that was responsible for development of pollen tubes. Over there I told you that this is an important mineral which we will study how it enables the formation of pollen tube. As soon as the eating up of the style tissue by pectinases and hydrolases begins, it is this complex which enables, which provides the stimulus so that the pollen tube extends towards the ovule and the pollen reaches to the micropylar end of the ovule. Now as soon as it reaches over there, what happens? We will see that, but before that please revise these terms. Pollen tube is being formed. The process is called siphonogamy. It is being guided by a chemical complex, this one, and this entire process is being done in order to enable the fusion of male gamete. Now you know that in the male gamete or in the pollen grain, there are two male gametes which lie within, not one. So two male gametes are going to combine with the female gamete. Now we are going to see what fertilization is all about, and when we see that, we come across two more terms. What are those terms? We are going to see and pile up the lesson, because this happens to be simple introduction to fertilization. What are the course of events? We see that with the help of a presentation. So the two male gametes, as soon as they reach the micropylar end, you know that at the micropylar end there is arrangement of synergids. These synergids enable the entry of two male gametes. So first of all, what happens? The first male gametes male gamete, not gametes. The first male gamete, it combines with the egg that lies next to synergids. The synergids enable its entry, and it is a chemically guided process. One male gamete combines with the egg, and this particular process is known as fertilization, but it has a name, that is generative fertilization, otherwise known as syngamy. So here we have the first male gamete out of the two undergoing a fusion with the actual female gamete, that is the egg, resulting into a zygote, which would later on develop to an embryo that we study as post-fertilization changes. We get a zygote. When the zygote is formed, the second male gamete, it goes to the polar nuclei within the central cell. Okay, two polar nuclei we had. Remember, it was a 7-celled 8-nucleate stage where the central cell had two polar nuclei. This is within the central cell. So this process is responsible for development of endosperm. Okay, now this endosperm, please remember, what is the result? As we had a zygote, over here we are going to have an endosperm which is responsible for providing nutrition to this zygote. Now in a way it is responsible for vegetative activity. So this fertilization is known as vegetative fertilization. Like the first one was termed generative, this is vegetative fertilization because it gave rise to endosperm. Other name for this vegetative fertilization is triple fusion. Now why it is triple fusion? As you can see, there are going to be 3 nuclei which would combine and a triploid would be formed. Triple fusion giving rise to a triploid endosperm, and hence the term triple fusion. Okay, so the triploid endosperm is formed as a result of the vegetative fertilization that has taken place between the second male gamete and 2 polar nuclei of the central cell, and as a result we get this endosperm. This endosperm is responsible for providing nutrition to the zygote. So you see one point, that after and only after the zygote is formed, then the formation of endosperm takes place, or rather I would say that triple fusion takes place. So in a way, angiosperms have a less wasteful fertilization process. One more point why they are so advanced. So we have this generative fertilization or syngamy, and we have this triple fusion. That is why the fertilization in angiosperms is known as double fertilization. This is a characteristic feature, so certainly it has a weightage from examination point of view. What is double fertilization? It is present in angiosperms. Why it is called double fertilization? Because there are two male gametes, they are going to combine with one female gamete as a result of the formation of pollen tube, the formation where the male and female gametophyte are brought together, which enables the fusion of gametes in two respective fertilization events. This is known as double fertilization. Now, what are the respective fertilization events? First one is syngamy, that is also fertilization. So, we have one fertilization. What is the second one? That is triple fusion. So, we have double fertilization that takes place in the angiosperms. So, this is all about the fertilization that takes place in the angiosperms when two gametes, both male and female, they come in contact with each other.